everyone. I'm Aaron Mertens coming to you live from INTERPHEX, New York City. And I'm here with my colleague, Paul Lopolito, who is a Senior Technical Services Manager. Paul has a technical talk Thursday at 10:30 discussing master soil selection for cleaning validation of a parts washer. Paul, would you like to introduce yourself and give us a sentence or two about your talk on Thursday?
Yeah, sure. Aaron, thank you for having me out here for the interview. I love attending INTERPHEX, and I try to get out here on a yearly basis, and glad to see it's back live, which is exciting. I'm a senior manager within the tech services group of STERIS, and I support process cleaning, clean and validation, contamination control for facilities around the world. And today, or actually on Thursday, I'm going to have a talk on master soil selection and clean and validation for parts washers. So I'm really excited to talk about that because it links laboratory testing I do a lot of testing in the laboratory in St. Louis with my team, and also the factory acceptance testing that's done in the STERIS facility outside of Quebec.
Oh, great. So this is really a collaboration, so to speak, between two different divisions or locations within the STERIS Corporation.
Yes, that
Great.
is absolutely correct. And
Very
that's one
nice.
of the things that we saw as kind of an interesting opportunity in the sense that our customers would schedule a factory acceptance test to assess the washer that they purchased in regards to design specifications and user requirements and the functional specifications on the washer, but oftentimes wouldn't be allowed to do a full cleaning study as part of it.
Okay.
And that's where the testing lab in St. Louis comes in, because we do a lot of cleaning evaluations to develop a cleaning procedure. And so by us working on the cleaning procedure and then sharing that information out with the factory, it allows that connection between the customer in the cleaning and the manufacturer of that equipment.
Right. So you're using laboratory studies and laboratory data to then prove out the effectiveness of a cleaning cycle, the cleaning equipment, the washers in the factory prior to customers accepting that equipment.
Yeah, that's correct, Aaron.
Great.
The thing that's interested, though, is that what we can do in a laboratory setting with chemists and physicists and biologists is test a wide range of samples,
Right.
from biologic samples to chemical samples to active pharmaceutical ingredients to even some highly potent materials. These materials really can't be shipped out to a factory to do a cleaning evaluation. And that's where kind of bridging the gap between a laboratory setting and the factory makes sense. And that's
Okay.
where developing a master soil comes in quite handy. We've seen a lot of customers take a look at their product portfolio and group them and identify a worst case cleaning challenge based on laboratory testing. But what we want to do from a master soil perspective is take a look at a soil that is a surrogate or representative of what soils that they would be producing on the equipment. And then having that surrogate be tested out in the laboratory to show that it is similar or harder to clean than their current portfolio.
Right.
And then that sample, that master soil, could then be utilized potentially as part of the factory acceptance test. So they can do coverage testing to assure the equipment is designed to get proper wetting of the parts, but also they can use a master soil that's safe and that can be shipped, that is going to be easy to work with from the operator's perspective to the environmental perspective, and use that as part of the cleaning challenge at the factory.
Right. So presumably you have some recommendations for a master soil or data or a process to help users start to develop or determine what a good master soil is for their cleaning process.
Yeah, and that's really going to depend upon what types of products that they're producing, and then using some type of risk matrix to kind of understand what makes sense to use not only as part of a factory acceptance test, but also as a representative soil when they go into their own clean and validation.
Okay.
So yes, we did evaluate quite a number of them, but they're generally very specific to the customer's applications on what makes sense. So
Right.
if you're manufacturing, say, a biological product, say a monoclonal antibody, you may say, okay, we want to also evaluate maybe tryptic soy broth, TSB. We may select maybe a different soy protein. We may select maybe something like xanthan gum, or maybe a sugar, something in which it's relatively inexpensive. It's a defined soil, so it's going to be consistent if you get it again. It's also not introducing any new hazards to your production equipment, so free of animal materials, maybe free of dyes, maybe free of lubricants, things that could present problems.
Right.
The last thing you want to do is introduce a soil into your equipment and then cause other issues, and we've seen that.
Okay.
And then the important thing with that is showcase that it's going to be similar in performance as your routine soil.
Okay.
And what that does, it could also help out in terms of streamlining your validation once you get the equipment in, because you can kind of test it out and you can run that as a surrogate for your product soils.
Well, this is an interesting concept. It seems to me that this is akin to using biological indicators to evaluate effectiveness of an autoclave sterilization process, for example. Would you agree to that?
It has some similarities to that. Yeah, because you're kind of you're testing out the unit and making certain that it can perform according to desired acceptance criteria.
Right.
Using a test article.
Okay.
And that test article is going to be showing a degree of resistance compared to the load that you place into the autoclave. So this is the same type of thing. You've identified a test soil that has similar performance or similar cleaning challenges as your product that you're producing, and you're evaluating that in a more controlled, in a deliberate way.
Right.
So it can have similarities to that, yes.
Okay. Well, I was thinking about this. I suppose that there is a consistent challenge with the master soil, but then are there specific parameters in the wash process that are controlled, or specific attributes of the wash process that are important as part of the test?
So yes, there are some critical steps within the cleaning process, and those can be easily controlled through the washer. So things like rinse time, rinse temperature, quality of the water used, the wash time and temperature and concentration of the detergent, the rinse times, even kind of rinse conductivity, so you can kind of monitor that. Some of the washers even have total organic carbon testing.
Okay.
And they have things like the drying cycles and air blows and even filtered air into the chamber. Some of those things can be looked at as part of laboratory testing. Some of those can even be challenged with a master soil as part of the testing as well. Again, a lot of this is to have be very comfortable with your information and your criticality of parameters as early in the process as you can, because the more investment that you have early on, the easier it is to go into commissioning of the parts washer when it arrives at the location of installation.
Okay.
And also the validation activities on the washer.
So am I understanding you correct that in the laboratory environment, you have equipment or capabilities to control some of those process parameters that you would have in the equipment being built at the other STERIS facility?
Correct.
Great. So that helps you develop your database to have these master soils.
That is correct.
Okay, very nice. Moving on, I'm shifting gears here a little bit. This sounds like a great approach to me. It seems like a good path forward. Do you have a feeling for how regulations or regulatory bodies are accepting this type of concept?
Yeah, that's a great question, Aaron. Every time that we look at approach to, I want to say, simplify a way in which we can leverage data, we always want to take a look and see what regulations are out there. The idea of grouping or bracketing or matrixing products is well documented in FDA Guide to Cleaning Validation, as well as the ICH, as well as Health Canada, World Health Organization. They talk about having a scientific approach to group or bracket products,
Right.
Based on a worst case.
Okay.
So identifying a surrogate of comparable cleaning performance or worst case cleaning performance is right in line with the regulations as they exist today.
Okay. You named off a number of regulations, global type regulations. Are these pretty well harmonized with this concept, or do some of them differ? Do we have some outliers with the regulations, or
No, so they align.
pretty well harmonized?
Pretty nice.
Okay.
Yeah. And actually, it was interesting reading the latest. There was a guide from the World Health Organization that talked about setting health-based exposure limits and a value in doing laboratory studies to support your cleaning cycle, and also the use of laboratory testing to support a worst-case test soil.
Okay.
Or worst-case product
Right.
for evaluation. So it seems to be right in line, and most of the guidance that I've seen they're aware of what has been previously published, and they're looking at reinforcing that and then highlighting a couple of items from that.
Right. If I'm understanding this correct, this is a concept that each company with their specific soils, their product residues, or what it might be, they're evaluating their specific residues to develop or find a representative master soil. It's each company, not just one across the industry, for example.
Yeah, this is the talk that is based on some research.
Okay.
So we did evaluate about, I think it was about 12 to 15 test soils from a wide range of industries that we have seen customers use as a master soil, and then applied kind of a risk-based approach to that, looking at availability, looking at cleaning performance. We looked at toxicity. We looked at environmental exposure, operational exposure, and then we even evaluated visible residue limit, which is kind of important, and I'll talk a little bit about that. So some of that information has been published in an article in Pharmaceutical Engineering on the same topic, leveraging a master soil for factory acceptance testing of a parts washer.
Okay.
And so customers or individuals can look at that and say, okay, this soil is relatively safe to work with, relatively available, easily shipped internationally or locally, is safe for the operators to work with, low toxicity, and cleans in a similar manner as our test sample. So yes, they could kind of look at that and say, okay, I'm making a biologic product, I'm going to use xanthan gum, here's some information on that from this literature. Mm-hmm. My recommendation is that's a good starting point, but it's always best to do some laboratory testing to compare that master soil selection to your current products that you're going to be producing, so that you have that in hand and says that this product is going to be very similar to the products that we're producing, and you have that scientific rationale for why you've selected that as a master soil.
Okay. Well, that seems like a lot of rationale to put into place. What would be the driver for a company to demonstrate the effectiveness and the purpose of a master soil?
Yeah, it's also an interesting question, Aaron. I mean, I think the driver is, and it kind of goes back to the whole rationale for the factory acceptance test, and that is really that you have a design in mind, you have your user requirements, you have your functional requirements. You've tested that out as part of the factory acceptance test. Now, the more complete that testing is, the more precise, the more clean those results are as part of those testing, it brings the work for the commissioning and the validation so much easier. So by having increasing that to a cleaning challenge makes a big difference up front.
Yeah.
Also, we see that sometimes that's overlooked early on because of the risk of sending a sample, the risk of doing that testing, so they bypass that. So they then introduce that as part of near the end of the commissioning. And so therefore they come into surprises in terms of how difficult it is to clean the residue once they actually are starting to do the cleaning procedure. And then it adds delays and cost associated with their commissioning and eventually could be their validation activities. Depends upon when in the process they do that.
Okay.
So as you introduce this early on and you do some laboratory testing to develop your cleaning procedure, then you can leverage that as you get into your validation work.
So it's an investment. You're putting the work in up front, putting the effort in the lab, bringing that information to the factory acceptance test, building a robust cleaning process, and saving yourself time and effort in the next phase of commissioning and qualification.
Aaron, you said it much better than I did. That was excellent.
Great. Well, the last question I have here, Paul, is what aspect of this concept will have the biggest impact moving forward? What do you see on the horizon with this concept of master soil?
I think I want to say maybe two things. One is the importance of design testing in terms of identifying your critical parameters with cleaning, and secondly, the importance of using that information in terms of evaluating, in this case, a parts washer at the factory, in which case you have the subject matter experts there. You're walking down that equipment, you're evaluating the coverage testing, you're evaluating the cleaning testing, and you're feeling very confident when that equipment comes into the site for use in production. Mm-hmm. Yeah, I think that's two of the things that I would think about up front.
Great. Well, this has been an excellent conversation. I feel like I've learned, increased my knowledge exponentially just having a quick chat with you, Paul. I will personally plan to attend your talk Thursday at 10:30 here at INTERPHEX. Again, thank you for your time. I appreciate your support, and I look forward to hearing more about this topic from you.
And thank you, Aaron. Great questions. And again, I'm also looking forward to my talk at 10:30. And again, if you can attend the talk, then it's also the information was published in an article in 2017 in Pharmaceutical Engineering, and I'd be happy to share a link to that.